Blood Transfusion Part 2. This current video is about blood components and the causes of acute blood transfusion reactions. Currently, available blood products are sourced from individual donations. Commonly used blood components are red blood cells, platelets, plasma, and cryoprecipitate. Red blood cells carry oxygen from the lungs to cells throughout the body for use in cellular respiration, attached to the hemoglobin protein in the blood, and they return carbon dioxide from the cells to the lungs so it can be exhaled. Red blood cells are transfused to patients with low hemoglobin or low red blood cell count, or when clinically significant symptoms of anemia are present. Transfused red blood cells must be ABO compatible with the recipient. The main function of platelets is to stop or prevent bleeding. Platelets are transfused to patients with low platelet counts or in certain cases of hemorrhage. When platelets were activated, they will change from the typical discoid shape to an elongated form, simultaneously activating the coagulation cascade. Plasma, the liquid portion of blood, contains all clotting factors, fibrinogen, plasma proteins such as albumin, and electrolytes. It is stored frozen and is thawed when there is a need to replace critical proteins for blood clotting and immunity. Cryoprecipitate is a portion of plasma rich in clotting factors, including factor VIII, 13, von Willebrand factor, and fibrinogen. This product is used to control massive blood loss, for example during trauma. Blood transfusions can save lives, but are not without risk. This means that undesirable or adverse reactions can take place, ranging from mild to life-threatening. These occur because there are varying degrees of mismatch between the recipient's and the donor's blood. Acute blood transfusion reactions most commonly present during the first 15 minutes of a transfusion. Many of the adverse effects of blood transfusions are mediated by the recipient's immune system. It occurs when incompatible blood products are transfused into a recipient's circulation, triggering a response from the recipient's immune system. Immune-mediated transfusion reactions include febrile non-hemolytic transfusion reaction, mild allergic reactions, anaphylaxis, acute hemolytic reactions. We shall discuss them one by one. The most common immune-mediated transfusion reaction is a fever without signs of hemolysis. This is called a febrile non-hemolytic transfusion reaction. The cause is thought to be the recipient's preformed antibodies attacking transfused donor white blood cells. Another factor might be that during the storage of blood units, white blood cells release cytokines that may provoke a fever when the unit of blood is transfused into the recipient. The next is an allergic reaction. It can range from a mild allergic reaction to life-threatening anaphylaxis, which will require immediate action. In mild allergic reactions to a blood transfusion, either the transfused blood contains immunoglobulin E that binds to the antigen from the recipient's blood, or the antibody is the recipient's own and binds to the antigen in the transfused blood. This happens when existing immunoglobulin E binds to its antigen and triggers the release of histamine from the recipient's mast cells and basophils. Anaphylaxis is a life-threatening allergic reaction that can occur after only a few milliliters of blood have been transfused. Usually, the antigen that triggers the anaphylaxis is not known. In the case of recipients with immunoglobulin A deficiency, it's thought that the presence of immunoglobulin A in the donor's plasma is the trigger. Acute hemolytic transfusion reactions is another life-threatening reaction. This is because as few as 10 milliliters of ABO or rhesus-incompatible blood can cause a substantial reaction. It can be intravascular or extravascular hemolysis. The most severe reactions involve an intravascular hemolysis. The donor red blood cells are destroyed by the recipient's antibodies while they are still inside blood vessels. Such reactions involve antibodies which lyse the donor red blood cells. Hemoglobin is released into the plasma and excreted in urine, turning the urine red in color. This is also known as hemoglobinuria. Other tissue factors released by red blood cell debris increase capillary permeability, leading to vasodilation, hypertension, fever, and disseminated intravascular coagulation, also known as DIC. In extravascular hemolytic reactions, the donor red blood cells are removed from circulation by macrophages in the spleen and liver. The macrophages destroy the red blood cells inside these organs. Because the extravascular destruction of red blood cells is slower and more controlled than intravascular hemolysis, the main symptoms of this type of reaction are fever and chills. Not all reactions arising during a blood transfusion can be attributed to the immune system, such as transfusion-transmitted bacterial infection. Bacteria can be passed on from the donor who is unaware that he or she has an infection at the time of donation. Infection may also occur after the blood has been donated. Bacteria can contaminate blood products while they are being stored. Macrophages engulf the bacteria. Cytokines are released and stimulate the brain to provide a response, like fever, swelling, pain, and erythema. No matter what kind of transfusion reaction happens, it can be dangerous. Therefore, early detection is important. You come to the end of acute blood transfusion reaction video.